So good morning and welcome back to my channel Biochemistry Quick Review and Guide. Today in this video I'm going to tell you about how to identify a carbohydrate from an unknown solution. So what are the different tests you are going to do sequentially. So you're just going to see that and try to understand so that you can find out what glucose or what kind of carbohydrate is present in that particular solution. So the first test in carbohydrate is Mollis test. Mollis test. First of all, you will do Mollis test. Mollis test. Mollis test. Mollis test detects presence of carbohydrate. Procedure and all these things I'm not going to discuss. I'm just going to discuss that flowchart scheme which you have to follow to uh, rule out what kind of carbohydrate is present in that particular solution. So first of all, you'll do Mollis test. Mollis test. If Mollis test is positive, if Mollis test is positive, if the Mollis test is positive, it means carbohydrate is present. It means carbohydrate is present. Carbohydrate is present. Okay, I'll write like this. Carbohydrate is present. And if it is negative, you'll see if it is negative, that means carbohydrate is not present. Carbohydrate not present. Okay, so if it is positive, carbohydrate is present. Next test, what test? The next test that we're going to do. Next, we are going to do iodine test. Iodine test. We want to find out what kind of carbohydrate is present. So if iodine test is positive, that will go like this. If iodine test is positive, it means that the solution contains polysaccharide. Polysaccharide. Like it may be glycogen or starch or any polysaccharide. Okay. And if the iodine test is negative, it means no polysaccharide. No polysaccharide. No polysaccharide. It means that it may contain monosaccharide or it may contain disaccharide, isn't it? Because carbohydrate is present. But only what we find here is that there is no polysaccharide. So it means it may be monosaccharide or disaccharide. So when it is negative, this is negative. Okay, this is negative iodine test. If the iodine test is negative, then we will do next. We'll be doing next test is Benedict's test. We'll be doing Benedict's test. Benedict's test is done to see the presence of reducing sugar. So Benedict's test. If Benedict's test is positive, what does it mean? Reducing sugar present. Reducing sugar is present. Reducing sugar present. So reducing sugar, it may be either disaccharide or monosaccharide. Reducing disaccharide, it may be reducing disaccharide. Reducing disaccharide like maltose and lactose, like maltose or lactose, or it may be reducing monosaccharides like glucose, it may be galactose, mannose, etc. Okay. Now, if Benedict's test is negative, if Benedict's test is negative, if it is negative, it means that there is no reducing monosaccharide or disaccharide. That means it may be reducing disaccharide. What is the reducing disaccharide? Non-reducing disaccharide is, it may be a non-reducing disaccharide. Okay, like sucrose, it may be sucrose, because sucrose is a disaccharide which is non-reducing. But we are not concerned about this right now. So now we've got reducing sugar is present. Now next is what we're going to find out is we will be doing in this. We'll be doing now next. We'll be doing Barfoed's test. Okay, Barfoed's test. Barfoed's test will help us to identify whether it is reducing monosaccharide or reducing disaccharide. So if it is positive, if Barfoed's test is positive, it means if the Barfoed's test is positive, it means reducing disaccharide. Reducing disaccharide. I'll write in short. It is reducing disaccharide. Okay, it may be reducing disaccharide. That means it may be maltose or lactose. And if it is negative, it means it is reducing monosaccharide. Reducing monosaccharide. It will be reducing monosaccharide. Okay. Now monosaccharide can be again of two types. Let me write here monosaccharide. This monosaccharide. Let me pick up from here. Let me start from here. Okay. Now we got monosaccharide. It may be monosaccharide. And again, monosaccharide can be of two types. Monosaccharide can be either it may be keto sugar or it may be aldose sugar. So to identify whether it is keto sugar or aldose sugar, you'll be doing what's the test? You'll be doing a test that is called Selivanov's test. Selivanov's test. I don't exactly what is the spelling. Selivanov's test. So if Selivanov's test is positive, it means what is that? It means reducing keto sugar is present. It means keto sugar. It means keto sugar is present. Keto sugar is present. And if it is negative, it means aldose sugar is present. Aldose sugar is present. Aldose sugar present. That means keto sugar. Example we have fructose. And aldose sugar we have glucose, galactose, mannose, etc. So this is how you are going to identify your unknown solution of carbohydrate. Okay. So simply follow this flow chart and you'll be able to understand and able to identify the monosaccharide, or the carbohydrate that is given in the solution. So thank you. So in the previous slide, Bendix test negative means that reducing sugar is not present. Bendix test was negative. Benedict's test. Okay. Benedict's test negative. Benedict's test positive. We have already seen positive means reducing sugar. Positive is reducing sugar. And negative means reducing sugar is not present. Reducing sugar not present. That means it may contain non-reducing sugar. So in this case, when the Benedict's test is negative, that time you have to do hydrolysis. You have to hydrolyze hydrolysis done in water bath, and after hydrolysis split the sample into two, and in one of the samples do Benedict's test, and in one of the samples, in the other sample you do Selivanov's test. Selivanov's test. Benedict's test, if it is positive, means that the non-reducing sugar that is present has been hydrolyzed. And Selivanov's test positive. Benedict's test positive will mean that it may be glucose. And Selivanov's test positive means that the solution contains fructose. So after hydrolysis, that means the solution contains sucrose, which after hydrolysis is converted into fructose and glucose. Therefore, Selivanov's test and Benedict's test are positive. That means this non-reducing sugar present here is sucrose. And in Barfoed's test positive, here you have seen when the Barfoed test is positive. When Barfoed's test is positive, it means that the solution contains disaccharide, reducing disaccharide. Now, how to identify this disaccharide? It may be lactose or it may be maltose. It may be lactose or it may be maltose. But how to identify? To identify this disaccharide, you have to do test that is called Osazon test. Osazon test. And in Osazon test, it will be found that this lactose will produce powder puff shaped crystals. They will produce powder puff shaped crystals. Whereas this maltose will produce sunflower shaped crystals. Sunflower shaped crystals. Which you can see in the microscope. So these are the characteristic osazons. By identifying these osazons, you can identify whether it contains lactose or maltose. Okay. And actually, osazon test is given positive by all reducing sugars. So these are the reducing disaccharides. And reducing monosaccharides like glucose, galactose, glucose, fructose, and mannose. They all give same osazon. That is, they give what is that called? They give needle-shaped crystals or broomstick appearance. Needle-shaped crystals. They give needle-shaped crystals or broomstick appearance. Why this glucose, fructose, and mannose they give same osazon is that because during osazon formation, the first and second carbons are involved. So since these structures differ from each other in the first and second carbon, therefore they give same osazon, because during osazon formation these two first and second carbons are masked. Therefore the osazon appears same in these three reducing monosaccharides: glucose, fructose, and mannose. Thank you.